Hi everyone. My name is Matthew. Welcome to this class. Today we are going to explore the pathophysiology involved in Parkinson's disease. We are gonna see all mechanisms related to the disease onset. We hope you enjoy it and, please, pause the video as many times you need. Our first question here is the major one. How does Parkinson's disease develop? Well, what happens is that some cells start to die in the brain. Particularly, there is a loss of neurons that use the dopamine in a specific area of our brain, called substantia nigra. However, this is not the only mechanism behind the physiopathology of Parkinson's disease or PD. We also have the involvement of other neurotransmitters and brain regions, and a widespread accumulation of an intracellular protein called alpha-synuclein. Please, take a look at the figure 1 where we can see a depigmentation of the substantia nigra, right panel, in a person with PD compared with a person without the disease, left panel. We can see here the macroscopical, inset, and transverse sections of the midbrain. Upon immunohistochemical staining for tyrosine hydroxylase, an enzyme involved in the synthesis of dopamine. When people are in the early stage disease, the loss of dopaminergic neurons is restricted to the ventrolateral substantia nigra, but becomes more widespread by end-stage disease. Looking at Part B, we can observe a hematoxylin and eosin staining of the ventrolateral region of the substantia nigra, showing normal distribution of pigmented neurons in healthy controls. Differently, in Part C, we can see a moderate pigmented cell loss in Parkinson's disease. And when we look at Part D, we can see a severe cell loss. In addition to the dopaminergic loss, another problem is the abnormal deposition of alpha-synuclein in the cytoplasm of certain neurons. Important to note that this deposition of alpha-synuclein happens in different brain regions. Looking at these images, immunohistochemical staining of alpha-synuclein shows the round intracytoplasmic Lewy bodies are more diffuse when we look at the arrow in Part E. Moreover, we can see granular deposits of alpha-synuclein in Part E, and in Part F. The images also depict deposits in neuronal cell processes, and extracellular dot-like alpha-synuclein structures in Part F, and alpha-synuclein spheroids and axons. Look at Part G. The accumulation of alpha-synuclein occurs in other neurodegenerative diseases, such as Alzheimer's disease. Lewy-related pathology, primarily comprised of alpha-synuclein, is present in a majority of autopsied Alzheimer's brains, and higher levels of alpha-synuclein in the cerebrospinal fluid of patients with mild cognitive impairment, and Alzheimer's have been linked to cognitive decline. In a landmark paper, Brock and colleagues first introduced the concept of spreading of pathology in the Parkinsonian brain. They argued that alpha-synuclein pathology gradually engages more brain regions as the disease progresses. In people with PD, the alpha-synuclein aggregation occurs mainly in cholinergic and monoaminergic lower brainstem neurons, in asymptomatic cases, known as Brock stage 1 and stage 2. In Brock stage 3 and 4, infiltrate-similar neurons in the midbrain and basal forebrain are predominant in those with motor symptoms of PD. In Brock stage 5 and stage 6, these infiltrate neurons are found later in limbic and neocortical brain regions with disease progression. The heterogeneity of Parkinson's disease is also underpinned by a complex pathophysiology. Examples of these pathways include alpha-synuclein proteostasis, mitochondrial function, oxidative stress, neuroinflammation. A progressive, age-related decline in proteolytic defense mechanisms in the aging brain might play an important part in the accumulation of alpha-synuclein proteostasis. Although it has been proposed that low levels of alpha-synuclein are normally present in mitochondria, the accumulation of the protein inside mitochondria leads to mitochondrial complex deficits and oxidative stress. Impaired degradation of specific functional proteins seems to have a role in the defective clearance of damaged mitochondria function. Thus, the activity of the mitochondrial complex is reduced in several tissues isolated from patients with Parkinson's. Because of mitochondrial dysfunction, oxidative stress is increased in the brain tissue of patients with Parkinson's. But it is debatable whether it occurs early or late during the demise of neurons disease. Nigral dopaminergic neurons have been suggested to be particularly vulnerable to metabolic and oxidative stress, for several reasons. Neuroinflammation is probably an essential contributor to pathogenesis as well, but maybe not the initial trigger. Looking at the motor cortex circuitry, PD involves a decreased dopaminergic transmission in the motor region of the striatum and increased inhibitory output activity of the basal ganglia. The basal ganglia are a group of subcortical structures, which have important functions in the control of actions and goal-directed behavior. Basal ganglia include striatum, globus pallidus, subthalamic nucleus, and substantia nigra. These circuits are anatomically characterized by a strong convergence of cortical input onto relatively few subcortical output neurons, and back to the cortex, suggesting a filter-like function. The motor circuit consists of corticostriatal projections from the primary motor cortex, supplementary motor area, cingulate motor cortex, and promoter cortex, terminating on dendrites of striatal medium spiny neurons. Looking at this image, the hyperdirect pathway directs glutamatergic connectivity from the motor cortex to the subthalamic nucleus. The globus pallidus internus and the substantia nigra pars reticulata are the two main output nuclei of the basal ganglia, and project to the brainstem and ventrolateral thalamus. The striatal projections to these output nuclei are divided into direct and indirect pathways. The direct pathway is a monosynaptic connection between medium spiny neurons that express dopamine D1 receptors and GABAergic neurons in the globus pallidus internus and the substantia nigra pars reticulata. The indirect pathway originates from medium spiny neurons that express D2 receptors, which project to the globus pallidus externus, and reaches the globus pallidus internus via the subthalamic nucleus as a glutamatergic relay. Through these two pathways, the striatal dopaminergic tone regulates the GABAergic output activity of the basal ganglia. As indicated, Parkinsonism is associated with changes in these relays. Indeed, nigrostriatal dopamine deficiency has opposing effects on the direct and indirect pathways. Although D1-mediated direct pathway activity becomes reduced, D2-mediated indirect pathway activity increases, resulting in a strong increase in the firing rate of GABAergic basal ganglia output neurons, which over-inhibit downstream thalamocortical and brainstem areas. More than that, another discussion is whether Parkinson's should be called a disease or a syndrome, as we have already seen. A multitude of mechanisms could underlie the final clinical expression of the complex disorder, that is Parkinson's disease. This list shows some of the mechanisms and pathophysiological basis that might be involved in the expression of clinical signs, including genetics and epigenetics, alpha-synuclein dysfunctions, mitochondrial dysfunction, neuroinflammation, gut microbiota, amyloid and tau deposition. Authors have suggested that PD is a heterogeneous syndrome, defined by a variable collection of signs and symptoms that manifest to differing extents within individual patients. In this sense, we already know that several neurotransmitter systems are affected, including the dopaminergic one. However, it is important to highlight that the noradrenergic, the cholinergic, and serotonergic pathways can be involved, among others. The involvement of different neurotransmitters may possibly express different symptoms. These possibly drive abnormal central brain network activity, which results in the clinical expression of the highly heterogeneous Parkinson's syndrome. The clinical phenotypic variations of Parkinson's represent the consequence of widespread brain and peripheral Lewy body pathology, and not a single neuronal structure, such as the substantia nigra or isolated loss of the dopamine neurotransmitter system. This concept of a multi-neurotransmitter, multi-organ, including brain and peripheral nervous system, disorder, is now well established in the literature, with supportive pathological and biomarker-driven projects. However, we are conscious of the fact that a robust evidence base for PD, as a multi-system and heterogeneous disorder, is lacking. Finally, let's see a little bit about the diagnosis of Parkinson's. Clinical diagnosis and natural history of Parkinson's disease are clinically defined by the presence of bradykinesia, as the slowness of movement and a decrement in speed, and at least one additional cardinal motor feature, rigidity, or rest tremor, as well as additional supporting and exclusionary criteria. The onset of motor symptoms is usually unilateral, and asymmetry persists throughout the disease. Well, clinical diagnosis looks simple in cases with fully developed classic motor features. However, early in the disease, error rates for a clinical diagnosis can be as high as 24% even in specialized centers. Accuracy of a clinical diagnosis can be improved using standard clinical criteria, such as the UK Parkinson's Disease Society Brain Bank criteria. This is the most used diagnosis criteria since 1992. In addition to the cardinal motor features, most patients with Parkinson's disease also have non-motor symptoms. It is also important to highlight here the need for diagnostic tests and biomarkers, to enhance diagnostic confidence in early disease, or to eventually diagnose Parkinson's disease in its prodromal stages. In summary, in this class we discussed that Parkinson's disease or PD is a heterogeneous syndrome defined by a variable collection of signs and symptoms. The physiopathology of Parkinson's disease is complex, and involves several brain areas and systems of neurotransmitters. Several processes can be involved in the physiopathology of PD, including the alpha-synuclein proteostasis, mitochondrial dysfunction, oxidative stress, and neuroinflammation. Thank you for watching this class. Remember to answer the quiz, it will help you to memorize the lesson content. Next class, we will talk about motor and non-motor symptoms in people with Parkinson's disease. See you.